Hello everyone, I am Anuja Shrivastava and I welcome you all in class 8th biology class. Today we will study about the third part of second chapter in which we will study about the artificial vegetative propagation. Artificial vegetative propagation means the plants are growing by the artificial means. So artificial vegetative propagation, it can be done by the three ways. First is the cutting. Cutting, that means the stem of one plant is cut and that stem is planted in the soil and that plant is growing into the new plant, and you are observing this way very widely. You have seen these things in the rose. Suppose this is the plant of rose and we cut this stem over here. This stem is cut from here and this stem is going to plant in the another soil. So this stem is going to grow into the new plant. That means the cutting way is also very helpful for growing the new plant, and this type of method is utilized in rose and sugar cane. Let's see the another method, and the name of that another method is layering. In the layering process, what is going to be performed here? The plants are emerge out from the soil to the upside. The soft branches of the plants, the newly soft branches of the plant, they are going to fold in this way. The newly branches fold and it is going to sow underground stem, and here one attachment which is known as a peg, that is there so that the root branch cannot come out from the soil, and the upper part that is going to be upside. That means the branch is going to dug in the soil, but the upper part of plant is the upper side. Now after timing, what happened? The roots are going to grow here. These are the new roots. The new roots are going here, and when these new roots are going here, at that time cut this branch from here and this branch now plant in the another pot so that the new plant is going to emerge out. And this type of process occur in the mostly ornamental flower like jasmine. So the next process of artificial propagation is the grafting. In this grafting way, most of the new types of plant can be germinated, and this type of grafting is mostly occur in mango, lemon. It also occur in rose. So how this grafting is performed? Here take one plant and cut that plant slightly like this, so that the plant which is in the soil, that part of plant is known as the stalk, and the part of plant which is now cut from the main plant, that is known as the scion. Now what happened? This scion is going to attach on the stalk of the other plant and tied it tightly with thread or with any other way. so that these two plants become one and grow as a one new plant. And in this way we can germinate the different varieties of mango, lemon and rosette. That's why this grafting method is performed. Now let's see the advantages and disadvantages of vegetative propagation. Advantages means how these vegetative propagation is useful, what are the merits of this vegetative propagation. So this method is very easy, rapid and cheaper. Because you have already studied about the vegetative propagation and you are knowing that in this process there is no cost of money, easily the things are going to be performed and new plants, we are getting the new plants. And the second one is advantages, the desirable characters of plants can be prevented. Now these are the two most important advantages. Let's see the disadvantages. Disadvantages, that means the demerits, that means whatever the problems are there. So if the parent plant have any disease, so that disease is going to transmit into the new plant. That means if parent plant is diseased, that plant is going to transmit to the another new plant. One more important thing, that this undesirable, unwanted character cannot be removed. That means if any unwanted character is there which we don't want in the next plant, so that character cannot be removed or cannot be eliminated. And when I told you that if parent plant is diseased, so already the new plant is going to be diseased with the same disease. Now one more important topic, that is the tissue culture, in which the plant is going to grow in laboratory. This is also the artificial method of the tissue culture. Now how this tissue culture is going to be performed. So in this method what happened, the tissues from a plant, the tissues, a small plant can be taken and take one tissue from that plant, that is tissue from X plant. X plant, why? Because that was the plant. That means they had taken the tissues. Now what that tissues is going, that tissue is put into the nutrient rich medium. When this tissue is put into the nutrient rich medium, the number of cells are going to they are going to divide number of times and a mass is formed. That mass is known as the callus. Now what happened after the callus is formed, that callus is again transferred into the different nutrient rich medium, and when this callus is again differentiated, the roots and the shoot is germinated. When this root and shoot is germinated, at that time that part is taken out and now the plant is ready to transfer and ready to plant in the soil, and in this way the desired character can also be formed. So this is all about the tissue culture, and the artificial means of vegetative propagation. So watch the video. Till then, bye bye. Thank you.